Hello and welcome back to Neuro Insights. I'm Rittika, and in this episode, we are examining neuroinflammation's dynamic role in Parkinson's disease, where immune responses in the brain can both protect and exacerbate neuronal loss. Joining us is Kanak Chaudhary with a Master of Science in Biotechnology, whose background equips her to bridge cellular mechanisms and therapeutic innovations. Kanak, it's great to have you back. Let's begin with the question of outlining the biphasic nature of neuroinflammation in Parkinson's, focusing on how alpha-synuclein aggregates trigger initial microglial activations.
Thank you so much, Ritika, for having me in the podcast. If we talk about the biphasic nature of neuroinflammation in Parkinson's, it reflects both its protective and damaging roles. Early on, the alpha synuclein aggregates act as danger signals, triggering microglial activation. In this initial phase, microglia clear the debris and secrete trophic factors which can be neuroprotective. Also, the aggregates persist and the activation becomes chronic and maladaptive, leading to a sustained release of pro-inflammatory cytokines. Also, the oxidative stress and ultimately contributing to dopaminergic neuron loss in the substantia nigra region.
Right, that's a very strong foundation, I must say. And now I believe we should probe more into microglial phenotypes. So, microglia exhibits M1 pro-inflammatory and M2 anti-inflammatory states, as we know. And I want to ask that what are the nuance shifts that occurs in these phenotypes during Parkinson's progression, and how do single cell RNA sequencing studies reveal heterogeneity in their responses to mostly the dopaminergic neuronal stress?
Yes, so microglia doesn't exist in only the two rigid states. Instead, they shift along a spectrum. And during Parkinson's progression, you often see a tilt from transient M2-like repair responses toward the prolonged M1-like pro-inflammatory activity. Also, the single cell RNA sequencing has been particularly revealing it, which shows that the heterogeneous subpopulations of microglia coexisting is there, and some upregulating interferon pathways and other showing that the metabolic reprogramming towards glycolysis is also there. This diversity underscores that not all microglia respond uniformly to the dopaminergic neuron stress, and targeting subtypes rather than broad suppression may hold the therapeutic promises.
That was extremely illuminating. Now, moving on to astrocytic involvement in it. So, astrocytes, I believe, are very overlooked most of the time, and they mostly modulate inflammation via cytokine release. So, how do they interact with microglia in the substantia nigra, and what cutting edge optogenetic manipulation demonstrate about their role in specifically amplifying or resolving alpha-synuclein induced inflammation?
So, astrocytes are increasingly recognized as active regulators of neuroinflammation. And in the substantia nigra, they engage in crosstalk with microglia astrocytic cytokines like interleukin 1 beta, and that can amplify the microglia activation, while astrocytic release of anti inflammatory factors can dampen it. And if we talk about the cutting edge one, the cutting edge optogenetic tools have shown that stimulating
Inflammation.
so astrocytes are increasingly recognized as active regulators of neuroinflammation. And in the substantia nigra, they engage in crosstalk with microglia astrocytic cytokines like interleukin 1 beta, and that can amplify the microglia activation, while astrocytic release of anti-inflammatory factors can dampen it. And if we talk about the cutting edge one, the cutting edge optogenetic tools have shown that stimulating the astrocytes can actually escalate the inflammatory cascades in the presence of alpha-synuclein protein, while silencing them can reduce microglial hyperactivation. So the astrocytes act as amplifiers or brakes, and this dynamic role is key to how inflammation evolves in Parkinson's disease.
Right. So that was a very compelling theory. And let's talk about therapeutic targeting, which is the natural flow of the topic. So, inflammasome activation, particularly NLRP3, is a hot topic. Can you discuss how mitochondrial derived damage associated molecular patterns, also known as DAMPs, fuel this in Parkinson's, and what phase specific inhibitors show in preclinical models using human iPSC derived brain organoids?
So the NLRP3 inflammasome is a central player here. Like the mitochondrial dysfunction in Parkinson's release these dams, such as mitochondrial DNA and reactive oxygen species, which serves as potent activators of NLRP3. This drives the interleukin 1 beta release and the neuroinflammation. In preclinical studies, especially in iPSC-derived brain organoids carrying Parkinson's mutation, phase-specific NLRP3 inhibitors show promises. In early inhibition prevents chronic inflammation without compromising the initial protective immune responses. And I think this gives us a more nuanced therapeutic window to target inflammasome activation without bluntly suppressing the immune system.
Right, there are several key challenges there. And moving on to genetic and environmental links, which I think is a very central topic. So, mutation in LRRK2 and Parkin genes heighten inflammatory susceptibility. So, how do these alter toll like receptor signaling? And what epigenetic reprogramming like age-tag inhibitors might mitigate chronic neuroinflammation in the at-risk population, do you think?
Okay, so if we talk about the mutations like a mutation in gene like LRRK2 and Parkin heighten the hyperbolized ways by altering the innate immune signaling. And LRRK2 mutations, for example, um aggregates the toll-like receptor pathways, priming the microglia for overreaction. And Parkin mutations reduce the clearance of damaged mitochondria, leaving more dams to perpetuate the inflammation that exists over there. And on the epigenetic side, the histone deacetylase uh, and the deacetylase inhibitors have shown that reprogramming inflammatory gene expression can dial down chronic microglial activation. This combination, understanding genetic predispositions while using epigenetic tools to reset immune tone, could help protect at-risk populations before neurodegeneration advances more.
Right, that was certainly many intriguing connections. So, the blood-brain barrier's permeability changes allow peripheral immune infiltration. So, what advanced proteomics reveals about systemic inflammation contribution to Parkinson's, and how do you think gut microbiota modulation via fecal transplant influence central neuroinflammatory cascades.
As Parkinson's progresses, the blood-brain barrier becomes leaky, that allowing peripheral immune cells to infiltrate and fuel the neuroinflammation in an above manner. And the advanced proteomics has mapped systemic inflammatory markers like complement proteins and acute phase reactants that correlate with disease severity. And at the same time, the gut microbiome has emerged as an peripheral driver. These viruses can generate the metabolites and microbial amyloids that reach the brain and influence the inflammation more. And fascinatingly, the fecal microbiota transplants in animal models have shown a normalization of neuroinflammatory markers, suggesting that the gut modulation could be a viable preventive or adjunct therapy for the same.
Right, so looking forward, AI driven network analysis of inflammatory pathway is emerging at quite a lot of speed. So, how might multi-omics integration predict inflammation trajectories in Parkinson's? And what ethical consideration arise in using such models for early intervention strategies, do you think?
So the AI-driven network analysis is giving us a systems level view of inflammation by integrating multi-omics approach, that is the genomics, transcriptomics, proteomics, and metabolomics. Machine learning can predict how these inflammatory pathways evolve in individual patients and even forecast when protective responses will flip into damaging ones. The potential for early ones, the personalized intervention is huge, and the key challenges are just as significant also. The data privacy, ethical use of predictive models, and ensuring these tools don't create inequities in access. The goal should be, I think, goal should be the responsible integration of such things, where AI supports clinicians in tailoring the interventions without replacing the human judgment.
Right. So, Kanak, you've indeed proven yourself to be a nuanced explorer of neuroinflammation and Parkinson's. Thank you. And for the listeners, stay tuned for the next episode.